Stage is yours.
Thank you. Well, there. Hello, everybody. My name is Pedro Salaberry. I'm a PhD student from Argentina, and today I'll be presenting our project titled Integrating Gene Expression Profiles with Non-Coding Somatic Mutation Analysis to Identify Key Regulators of Metastasis in Triple-Negative Breast Cancer. This subtype of breast cancer is characterized by the lack of 3 molecular markers, ER, PR, and HER2, and represents 15% of all breast ductal adenocarcinomas. TNBC has higher rates of metastasis and a poor prognosis relative to other subtypes, making a need out of the identification of new molecular targets for therapeutic intervention. With an interest on TNBC metastatic ability, we started off with data from a previous study in which 4 sublines of the TNBC cell line MDA-MB231 obtained from lymph node and lung metastasis were injected into nude mice, and primary tumors were surgically resected to evaluate metastasis and perform RNA-seq. These sublines, mainly isogenic, only differ in their metastatic ability, which comes at handy to identify regulators of this process. From this data, we perform a differential expression analysis using the well-known DSIC2 package for each possible pair of sublines, and we obtain differentially expressed gene signatures. Furthermore, we retrieved a breast cancer regulatory network through the Arachne Networks package, built from reverse engineering on transcriptomic data of TCGA breast cancer patients. Both inputs are necessary for the Viper algorithm to perform the differential activity analysis. Viper, for virtual inference of protein activity by enriched regulin analysis, uses regulin structures, that is to say, the mode of action of master regulators over the targets, to infer changes in the activity of those regulators, interrogating the expression levels of the targets. For example, if a master regulator upregulates genes found to be overexpressed in the analyzed contrast, it is considered differentially active in a positive way, whereas if the opposite occurs, it is considered differentially active in a negative way. If its targets are randomly distributed in the gene signature, it is not considered differentially active. This led us to a list of 354 significant regulators differentially active in at least one of the analyzed contrasts. Afterwards, we focused on non-coding somatic mutations registered in TNBC. We identified active promoters in breast samples using Phantom 5 cage data, selecting regions of interest above a certain score. We aligned those regions with registered TNBC mutations and filtered them by a functionality score. This score is an indicator of functional relevance predicted from a pool of known mutations using as sources their interest-specific conservation, associated histone modifications, transcription factor binding sites, and open chromatin states. From COSMIC and PCAWG databases, we selected those considered deleterious, finding 2,108 high-score TNBC mutations in active promoters of breast samples, most of which come from the PCAWG database. Finally, in order to integrate both results, we applied network propagation, a method based on the guild by association principle. If we select nodes of a network based on a certain feature, heat will spread throughout the network until convergence, allowing us to identify active subnetworks. Using the string database, we generated a protein-protein interaction network of the 354 master regulators and their 500 first shell interactors. After selecting genes containing high-score TNBC mutations and propagating that feature throughout the network, we filtered it to obtain an active subnetwork with the 10% hottest nodes. We functionally characterized it by means of the reactome database, identifying 2 protein complexes among other genes with recurrent high-score TMBC mutations. The first one was the COP9 signalosome, a protein complex involved in the ubiquitination-like process denatylation, enriched in terms like recognition and repair of DNA damage and clathrin-mediated endocytosis. The second one was the Swiss NIF complex, which is involved in chromatin remodeling, enriched in terms like methylation of histone arginines and interaction with RUNX1, a known TMBC master regulator. Currently, we are working on incorporating other functionality scores such as CAT and DeepC to the analysis. Furthermore, we plan to evaluate a few strong candidates within these key regulators to test their effects on TMBC metastatic ability and the consequences of the related mutations on promoters' activity. We believe this work presents an effort to show the potential application of this method to identify new markers for cancer detection and therapeutic targets, with the possibility to extend this pipeline to other types of cancer. We want to take a moment to thank the organizers of this event for allowing us to share our project. Thank you.
Thanks so much, Pedro, for this nice work and presentation. We can now get some questions. You can write your questions in the Q&A session or in the chat box as well. And also, if like, you can raise your hand so that we can give right now, I guess we have, no, yeah, okay. You can raise your hand so that we can give permission for you to speak. Okay. I gave the permission right now, me too.
Yeah, so sir, is there any microRNAs that has been signified in your studies?
Great question. Because of the way in which we created and generated these protein-protein interaction networks, microRNAs were left out of the analysis. But that is something that we want to incorporate in future analysis using other types of molecular networks.
Okay, I guess we have one question in the Q&A. You can check them out, Pedro, if you like.
Yes. I will read them out loud. The first one says, I would like to know if these genes identified in your study were previously known relevant in breast cancer. And yes, both protein complexes identified were found to be present in many in the breast cancer and even triple negative breast cancer literature. So yes, they were of interest because of that. But what comes to us as something really new is the identification of these regulatory mutations, of course, that we want to further analyze. And the second question says, thanks for your presentation. I was curious if you also tried other network construction tools besides Arachne, and what were the reasons to choose Arachne? Thank you. Okay, yeah, we did not try other network construction tools, but we want to in the future. And the reason for choosing Arachne was basically because it was used as a tool previously with Viper, this algorithm to infer protein changes in the activity of proteins. So yeah, basically that was the reason.